Baik, Bapak Ibu, kita mulai webinarnya. Selamat pagi Bapak Ibu semua. Assalamualaikum warahmatullahi wabarakatuh. Selamat datang di seri webinar Lab Sekuensing BRIN yang kedua, yaitu dengan topik Trends and Application of eDNA in Barcoding Monitoring. Webinar hari ini diselenggarakan oleh Direktorat Pengelolaan Laboratorium, Fasilitas Riset, dan KST, bekerja sama dengan Pandu Bioscience dan juga Illumina. Perkenalkan Bapak Ibu semua, saya Zahra Noviana dari Pusat Riset Mikrobiologi Terapan BRIN yang bertugas sebagai moderator untuk webinar hari ini. Jadi webinar hari ini kurang lebih akan berlangsung selama 2 jam dan kita akan ada 2 pembicara. Yang pertama ada Pak Rifki Sadikin dari Lab Sekuensing BRIN yang akan memberikan paparan terkait layanan ELSA untuk lab sequencing. Kemudian nanti ada pembicara yang kedua ada Mr. Terlibu, jadi Agri-Genomic Segment Marketing Manager for Asia Pacific, Japan, Middle East, and also Africa regions, yang nanti akan memberikan paparan terkait trends and application of eDNA in biodiversity monitoring. So we will have 2 speakers. Pak Rifki will present in Bahasa, and then later Mr. Cherilu will present in English. So we will switch between Bahasa and English for today seminar. Jadi untuk kurang lebih seperti itu, saya sekarang ingin mengundang dulu Pak Rifki untuk memberikan paparan terkait layanan ELSA Lab Sequencing, Pak. Untuk Pak Rifki dipersilakan.
Baik, Bu Zahra, terima kasih. Kali ini mungkin singkat aja ya, 15 menit diseminasi terkait ELSA dan juga program ke depan terkait dengan fasilitasi sequencing. Baik, saya mulai. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, Bapak Ibu semua, peserta webinar Lab Sequencing kedua. Perkenalkan, nama saya Rifki Sadikin, Kepala Pusat Riset Komputasi. Saat ini juga mengelola lab sequencing bersama teman-teman manajer lab ya. Di sini misalnya manajer labnya Bu Anik sebagai manajer lab sequencing, kemudian ada Bu Zahra yang sekarang moderator sebagai manajer ekstraksi, kemudian ada Syam sebagai manajer bioinformatika. Jadi untuk kali ini saya hanya share dua topik, yaitu terkait ELSA sequencing. Kemudian ke depan kita akan buka program sequencing dengan proposal. Jadi berbasis proposal untuk yang topik-topik seperti WGS, eDNA, dan lain sebagainya. Saya share dulu terkait dengan ELSA. Jadi untuk lab sequencing ini, mungkin langsung saja terkait dengan facilities ya. Facilitiesnya itu saat ini lokasinya ada di lantai 4 gedung genomik, lab genomik dan lingkungan. Itu ada di kawasan Sains Teknologi Soekarno di Cibinong. Terlihat di sini 2 ruangan ya. Ruangan pertama adalah ruangan sequencing yang kita lihat ada alat-alat sequencing baik itu dari Illumina, ONT, yang Sanger Base ataupun yang MGI. Kemudian ada ruangan untuk library preparation. Sebenarnya kami juga ada ruangan untuk ekstraksi sedang dioptimasi ya, sedang diperbaiki ruangannya agar nanti bisa menerima juga sampel dalam bentuk raw. Dalam hal ini sekarang sequence lab ini ada 3 lab operator OA sumber dayanya ya. Ada ini masih yang dulu nih. Untuk lab manager kita ada Bu Anik, ada Bu Zahra sebagai bagian ekstraksi dan Syam sebagai bioinformatik. Jadi memang dikelola supaya kita bersama-sama dengan periset-periset yang ada, kemudian ada tenaga dedicated untuk melakukan sequencing, library preparation ataupun ekstraksi. Sedangkan untuk ajuannya, alat-alatnya ya, alat-alatnya itu ada applied biosystem. Ini dipakai untuk sanger DNA sequencing biasanya untuk identifikasi. Kemudian kita ada platform long reads yang Oxford Nanopore, ada kurang lebih 4 minion MK1C, kemudian ada Prometheon. Untuk Illumina cukup lengkap ya dari MISEC, NEXEC, NOVASEC sampai iScan yang minggu lalu diperkenalkan. Untuk alat-alat Illumina ini pada tahun ini kesemuanya sudah di maintenance sehingga bisa dipakai dan tahun ini termasuk bahan akan disediakan di lab sehingga tidak seperti tahun kemarin di mana alat-alat yang berbasis Illumina itu sistemnya apa pemakaian ya bahan-bahan dari pengguna. Kalau sekarang kami coba untuk mensupport juga bahan-bahannya. Sedangkan dari platform MGI kita baru punya satu MGI 200. Nah, untuk layanannya sequencing sendiri kita ada sequencing berbasis Sanger, MISEC, NEXEC, NOVASEC, dan lain-lain. Sehingga nanti kalau untuk aplikasi itu memang dibuat seperti ini. Kalau mau lihat ini ada laboratorium pusat sequencing, eh, nanti bisa mengajukan berdasarkan alat ya. Dan nanti tujuan sequencingnya bisa ditulis di file yang disubmit ketika mensubmit layanan, sehingga nanti penyelia bisa melakukan pengecekan apakah layanan tersebut bisa dilakukan. Karena kan yang bergantung terhadap adanya consumable atau tidak, kalau bisa dilakukan, kapan dilakukan, dan lain sebagainya. Jadi di sini memang kita tidak buat perlayanan. Jadi kalau misalnya ada kebutuhan apa eDNA, ada kebutuhan mikrobiom, ada kebutuhan targeted sequencing atau whole genome sequencing, tolong langsung disebut di file yang nanti harus diisi ketika ingin melakukan pengajuan. Jadi ketika ingin melakukan pengajuan bisa dilihat di sini di berkas layanan ada SOP di sini bisa dilihat ada juga formulir-formulir yang harus diisi. Setelah itu nanti dari penyelia akan mengecek apakah ajuannya bisa dan seterusnya. Model pembayarannya ada dua, yaitu melalui PNBP ataupun dengan Elsa Point. Dan kami sedang mengusulkan agar Elsa Point-nya juga bisa melalui metode urunan, namun ini masih proses ya. Kemudian yang terakhir mungkin yang akan saya share yaitu terkait bagian program tahun ini yang rencananya merupakan bagian dari kegiatan koleksi dalam akuisisi data hayati ataupun non hayati. Jadi salah satu kegiatan yang kami usulkan itu adalah terkait dengan penambahan data sequence nucleotide ataupun struktur molekuler ke dalam dataset repositori ilmiah nasional. Jadi ini yang kami coba untuk menjadi salah satu kegiatan tahun ini. Untuk fasilitasinya adalah sampel-sampel yang berasal dari Biodiversitas Indonesia, kemudian eh, kegiatan fasilitasi layanan yang di diusulkan itu ada whole genome sequence untuk mikrobial, kemudian ada untuk hewan dan tumbuhan, dan juga eDNA dan juga mikrobiom. Tentunya ini berbarengan dengan kegiatan yang persiapan ya yang di lab sequencing, eh, termasuk ruangannya dan lain sebagainya. Sehingga nanti hasilnya adalah bisa menambah data sequence nucleotide dan struktur molekuler di BRIN. Nah, tentunya ini bentuknya nanti kita akan melakukan pengumuman pengumuman open call proposal bersama dengan kegiatan yang di koordinatori oleh Direktur Koleksi Ilmiah. Bentuknya pengumuman lebih lanjutnya mungkin nanti di series berikutnya terkait dengan ELSA berbasis proposal yang salah satu yang kita usulkan untuk difasilitasi adalah whole genome sequencing untuk yang saya sebutkan di sini. Sehingga nanti metodenya lebih ke mengirimkan proposal, kemudian nanti kalau ada reviewernya setuju, maka itu menjadi fasilitasi yang diberikan, semacam grant, tapi bentuknya fasilitasi lab gitu. Nah, ini yang kami coba desain pada tahun ini supaya mendorong penambahan data terkait data sequence di Indonesia. Kemudian terkait terms and condition-nya masih dalam penyusunan ya, jadi nanti mungkin setelah lebih jelas akan kami bersama dengan kegiatan yang lain itu akan kami sebarkan aturannya dan juga batasan-batasannya. Jadi kurang lebih seperti itu yang ingin saya sampaikan, bahwa di lab sequencing saat ini kami sedang mencoba mereaktivasi seluruh alat sehingga bisa menerima sampel dan alat itu bisa digunakan. Mohon nanti kalau ada yang ingin mengajukan, tolong ajukan saja diskusi ke kami sehingga kita bisa follow up. Misalnya kalau ada kebutuhan consumables yang belum, nanti kita coba disampaikan ke Kedeputian Infrastruktur dan Riset untuk inovasi, riset dan inovasi, supaya nanti bisa memfasilitasi sequencing yang menggunakan alat-alat sequencing, termasuk topik hari ini, yaitu eDNA. Barangkali itu dari saya sebagai mungkin pengantar ya terhadap kuliah hari ini. Terima kasih atas perhatiannya. Saya kembalikan ke moderator, Bu Zahra. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Baik, terima kasih banyak Pak Rifki untuk paparannya terkait layanan sequencing BRIN. Dan juga tadi sempat dibahas juga untuk open call for proposal lab sequencing yang akan dibuka dalam waktu dekat. Mudah-mudahan bisa memberikan gambaran yang cukup jelas ya Bapak Ibu semua terkait kegiatan-kegiatan yang akan dilaksanakan terkait dengan platform sequencing yang ada di lab sequencing BRIN. Kemudian, so we will moving forward to our second speaker. I mostly work in the field of microbial ecology and diversity, so today's topic is really interesting for me. I think for those who work in similar field, you will find the topic interesting as well. So with this, I would like to introduce our second speaker, Mr. Cherry Lu, the Agri-Genomics Segment Marketing Manager for Asia Pacific, Japan, Middle East, and also Africa regions. So Mr. Cherry Lu has been working with Illumina for the past 7 years and have been engaging with different key stakeholders in the regions. And eDNA is definitely one of the key interest area, and Mr. Cherry Lu has been working very closely with different eDNA researchers in the regions, as well as some key eDNA societies. So Mr. Cherry Lu will be sharing on the trends and application of eDNA in his presentation and some specific examples on how eDNA is being utilized in some monitoring and conservation space. With this, I will give the opportunity to Mr. Cherry Lu to present his talk. Yeah, so Mr. Cherry Lu, yes, now the time is yours. Yes, yes, I can.
You can hear me well,
Hi.
right?
Yes, yes, yes.
Okay, cool. Let me share my screen. Indonesia slide. All right. I hope all of you can see my slide.
Yes, we could see it.
Okay, cool. All right. Selamat pagi, everyone. Good morning, and thanks. Before I start, I would like to thank BRIN and Pandu as well for organizing this webinar, and thanks Zara for the introduction as well. So yeah, today is really my honor and privilege to be able to share my experience as well as my learnings as well throughout the region. So a short introduction. My name is Terry. So basically, I'm the segment marketing manager covering agrigenomics segment. So in Illumina's context, we call it agrigenomics segment, which I believe my colleague Evgeny last week has shared a little bit more on the animal side, the livestock breeding side. That's why I think Rifki mentioned about the eye scan machine as well. So today the focus will be on eDNA, environmental DNA. And for me, I think I've been covering the region for quite a few years. Hence, as you can see, the region I'm covering, Asia Pacific, Japan, including Australia, New Zealand, Japan, Korea, and just last year, we included Middle East and Africa as well. Hence, there's a lot of really, really interesting applications or examples of how eDNA has really picking up the popularity. And we are seeing the trend is getting more and more popular. And I think that's why today I'm happy to be here to share with you guys some trends and applications of eDNA, maybe specifically some examples as well that we see in the region, in some countries, in the area of biodiversity monitoring. All right. So if you guys have any questions, just feel free to type in, and then I believe later I will have time to answer those questions. So to begin with, I would like to just to give an overview of the agenda of my presentation today. So a quick coverage. So probably some of you might be already aware or quite clear about what is eDNA, but I think it's still good for the benefit of the rest of the audiences or participants who are not really working or not yet working on the environmental work yet. So I think a brief introduction of what's eDNA, and subsequently a high overview of trends and applications of eDNA that we see, and then as well as some specific things or events that we see or initiatives that we see in the eDNA space in the region. So hopefully this will be something that I think is useful for you guys in Indonesia, and something that potentially you guys could kickstart as well. So I always like to use this slide as the opening of my eDNA presentation. So if you look at on the left-hand side, so the title of this image is actually turning back the tide, or I would say big wave over here. So if you look at the smallest wave there, the red color wave is actually COVID, which all of us have experienced COVID time, and then now it's kind of like the post-COVID time. And then we have the economic crisis, recession, and then of course the always ongoing climate change, global warming issues that everyone has been discussing. But I think one of the main thing that we always neglect, or actually, as you can see, the biggest wave at the back is actually the biodiversity collapse. So this is when biodiversity is the health of the biodiversity is kind of ignored, and nobody really kind of monitor what happened to our biodiversity, and due to urbanization, a lot of different factors that impact our environment and surrounding. So this, to me, this is kind of a very good image just to highlight or to bring out the importance of why biodiversity monitoring it should be the focus for every country globally, right? And if you move on to the right, it's actually one of the image that we get from that I've gotten. I plucked it out from United Nations World Conservation Monitoring website. So it's actually highlighting like actually the situation that we are facing actually currently in terms of the biodiversity situation, or health of biodiversity that we are experiencing now. Is basically we are seeing extinction of species of animals, or even for plants. And then this is actually like the curve is going down and down. So this is actually kind of highlighting or bring out the importance of how can we actually bring up that curve again, And then on the most right-hand side, the 6 particular actions or efforts are the recommended action items that we could do potentially by every country in order to really bring up that particular biodiversity curve so that we can get back the original state of the environment that we have, or the richness of the biodiversity that we have. So in short, basically, this is just to kickstart the eDNA topic on my presentation in highlighting the importance of the biodiversity to us, or to human population. So what is eDNA and what is meta-barcoding? So one of my favorite photos that I always like to use, because this is kind of like the easiest image that maybe everyone can relate, right? Just imagine you go to a forest, a national park, and potentially what you will have is trees, you have rivers, you have streams, you have water, and of course, you have soil, and of course the animals, right? So basically for eDNA, it stands for environmental DNA. So basically it's DNA that present in environmental samples. So like what I mentioned, such as from soil, from water, or even from air. So air DNA is one of the trending area that people are starting to look into, and basically with the similar approach. So of course, some of you might encounter. So basically we will say like environmental DNA, probably you'll see some publications nowadays, eRNA as well. So same thing, people are starting, instead of DNA, people are starting to look at RNA as well that are present from our environment, and trying to study what is the, how do we utilize RNA as well to really monitor. So of course, today we'll be focusing more on the DNA side. The RNA one will be quite interesting because it's something really in trend and new. And of course, probably some of you guys who handle nucleic acid or RNA before, you know that how sensitive and easily degradable RNA is. So a lot of different protocols that you might need to implement if you're handling with eRNA. So basically for eDNA, going back to DNA, traces of DNA released by living organism. So basically via their skin, as you can see, the feces, mucus, skin cells, organelles, as you can name it. So basically as they walk past, let's say an area, they would basically just leave traces of DNA from that particular, it could be fish, it could be animals, it could be birds. So if I were to give you guys an even easier context to understand what's eDNA, is just imagine that I believe all of you will be quite familiar with like human forensics. If you are watching like CSI shows from US, you potentially, if your hand touches the table, and then the forensic scientists will be able to swap it and get your fingerprint, or so-called your DNA from the table. So it's basically the same thing, but the only thing is just that the DNA samples, or the DNA so-called fingerprint, is coming from environment. Okay, so it's as simple as that. So what is eDNA and what's meta-barcoding? So this is just in explanation terms. Basically, it's a cell-free DNA present in natural environment. And for DNA, if comparing to RNA, DNA is way more stable. So generally, eDNA remains detectable for up to 48 hours. So this also explains why people choose to start with eDNA first. Why DNA, right? And from the past, traditionally, of course, I wouldn't say like eDNA is really something new, but because some people might have already done some work previously, but it's just that the approach or the technology that they utilize might not be as advanced or as robust. So nowadays we have like what Ricky have just shared on all the NGS machine that we have nowadays. So as compared to last time, eDNA already can be detected basically using PCR or qPCR, but that time is maybe only for one particular individual species, and then which is known as barcoding. So basically for meta-barcoding, basically you can, from one sample, you can detect multiple species. And on the fourth point, basically when you want to detect the eDNA or from a particular samples, what kind of species or organism that is present from that particular samples, for example, water samples. I mean, normally the standard procedure is using the amplification of conserved region, and that's specific to the taxonomic class of interest, and then subsequently use NGS to sequence it. All right, so hopefully these will give you guys a good understanding of what is eDNA and what is meta-barcoding. Because basically, every time you heard people talk about eDNA, most of the time I would say you hear people talking about meta-barcoding approach. So when we move into the meta-barcoding approach, we can split it actually into, when we talk about NGS, doing meta barcoding using NGS, there are two different ways that we could do it. So first one, as you can see here, is targeted sequencing, where people target or amplify the conserved mitochondrial region. And then it is a taxon-wide approach. And since it's a targeted sequencing, basically you do not really need a super robust instruments to do it. So having said that, you look at the right-hand side, in fact, all the Illumina benchtop instruments, even as small as an i600, will be able to do the work for you. So that's on the targeted sequencing approach. And then on the shotgun, and another approach, which is on the other flip side of the targeted sequencing, it will be shotgun metagenomics. So I believe some of you have heard about this term as well. So basically, as is being named, shotgun metagenomics, when you shoot something, it's just like you just shoot and then you try to aim for as wide as possible, right? So trying to target everything. So that is basically sequence all DNA or RNA, and basically depends on the objective of your project, depending on what you want to look at. So for people who doesn't really have maybe a good idea of which particular bacteria, microbes, or species that they want to look at, and they have a sample, let's say from a particular river, from a particular region, and then this could be one of the way that people will kickstart their project if they have that access to higher throughput machine. Because for shotgun metagenomics, you need at least a NextSeq machine from Illumina, or even if you have a NovaSeq, I think what BRIN has now, I think it will be a perfect system to do shotgun metagenomics as well. So basically, this is just a simple meta barcoding workflow. So again, from no matter what kind of samples you're handling, first thing, once you collect the samples, a lot of time, especially for environmental samples, you definitely need to do a lot of filtering. And then you concentrate the DNA that you have, then subsequently you do the extraction and the amplification, subsequently NGS. And of course, one of the most important part is also on the bioinformatics part as well. So I think currently there are actually quite a number of free pipeline that you can utilize, which I can share a little bit more later, which there's a pipeline from our Japan researchers as well, and something that you could utilize very easily and after your NGS steps. And also subsequently species identification and ecological analysis. So basically this is just a high level overview of the meta barcoding workflow. So it's not something super complicated. It's not something rocket science, super advanced. As you can see here, meta barcoding, the easiest way to understand is the targeted sequencing. Once you have the particular barcode that you know of, and then you can perform meta barcoding easily. All right, of course, every time for Illumina, probably some of you are aware, Illumina, we are short read technology. We provide short read technology. So there's always this discussion like, so a lot of people might think that, oh, the longer I can sequence is always better, right? So in this context, in the eDNA space or in DNA context, are short reads sufficient? So this is always the kind of the key questions that people always ask, and thus having a longer read will enable better resolution or better information or more information that I want to see. So to me, I wouldn't say it's a definite answer, yes or no. But of course, something that I would like to highlight from this particular slide is to bring out, there are some instances that longer is not always better, as you can see from the bottom left publication. So it really depends on a lot of different factors, that how you design the particular experiment, how you design your primers, and then that particular region, is it a very difficult to amplify region? So a lot of different factors might impact the outcome or the information that you get in the end, right? So that will bring me to my next slide, which is when, let me see if I can bring this down. I'm not sure if this is blocking the slide. Just give me a moment. Just put it, okay. So from the short is short read sufficient? That bring me to this this subsequent slide is how does meta barcoding work, and some key points that we need to be aware of. So basically, eDNA surveys are indirect observation, because basically we are getting all the nucleic acid, be it DNA or RNA, that is present, and then we try to identify what's there. And then of course, the veracity or the accuracy of the meta barcoding findings really depends on the methodology, because again, it depends on the different type of samples that you are handling, potentially as comparing to, let's say, if the soil sample that you are getting is coming from an area that is really open to everyone, a lot of people, or not just animal, but even human, like pass by and things like that, which might have a lot of so-called contaminants or a lot of unwanted stuff that you don't want to have in your sequencing data. So that will actually affect or impact your meta barcoding experiment. And of course, different processes like I mentioned just now on even the most upstream part, where you want to concentrate your DNA, even the DNA extraction part, or how you are doing your filtering part. Every process could varies and kind of impact the downstream of your project. So in short, on the fourth point here, I mean, every experiment definitely will need a careful, a proper validation to make sure that the approaches that you take results in something that is unbiased and is something that we really want to see. Right, and then also given the absolute truth of diversity, and then a lot of times, basically the environmental samples that you have might be unknown. So sometimes one of the approach that people use is to have a mock communities for validation, just to make sure that at least the protocol or the approach that you are using is non-biased, is unbiased, and is something that is valid and legit for you to get the final results, and it's a trustable results that you are getting. So this is just basically some of the questions. I won't go through the details, just some questions that you need to ask before you perform your meta barcoding, or you plan for your meta barcoding experiment. Like it could be something like what I mentioned on the samples representation. Is it in abundance, or is only a minute amount that you're expecting? Hence, you might need a lot more DNA. So sometimes when, let's say, the particular organism is present in a really low amount, so sometimes that's when you need to kind of like bump up the DNA that you need to do for extraction. And then sometimes when you don't get anything in the end, you need to really come back to all these different steps to ensure that everything is checked and everything is in place. And also one of the thing that I just want to highlight a lot of, it's a busy slide here with a lot of points. So just to mention some key points as well, like I mentioned a little bit on the control as well. So of course, current gold standard biodiversity assessment, I think a lot of people or researchers nowadays are still based on the very first observational kind of or eyeball kind of observation when they are accessing their environmental samples. So with this method, it's always come back with poor reproducibility. And hence this is why people are nowadays are moving to genomics method, and then not just traditional genomics method, but also moving to NGS, which is way more robust. So of course you need to have positive controls, and then of course you need to consider the degradation of eDNA, and basically it will fall back again to what kind of samples that you have, the environment that you have when you are getting your samples. So on and so forth on the positive control side. I think something similar on the negative control side. So again, a blank is not always appropriate, and then sometimes when you do amplification, there always be bias that happen, and that will again fall back to the questions that how optimized your primer is, right? Your barcode that you have. So these are some of the key consideration points, like even spike-in controls, and also replication is essential as well. So I believe some of you who are doing experiments, planning for experiments, designing your experiments, these are all the similar, I would say, strategy or precautions that you need to take, or consideration that you need to take. Okay, I won't go into detail for this one. And then I want to quickly move into the trend and applications of eDNA that we see. So this is something that really interesting, and I think this will be something that is maybe will be of interest of a lot of you in terms of how do we utilize eDNA in which particular area, and if I'm dealing with marine samples, what are the applications that people are doing around the world, right? So in general applications of eDNA, I would say the reason I keep mentioning about meta barcoding, I believe if you guys who are dealing with environmental samples, you are doing NGS, I believe most of the papers or publications that you find will be using meta barcoding approach, right? And then be it different kind of species that you want to look at. So this is kind of like the so-called, I would say, like the unofficial gold standard approach that people are using now. And then of course, Illumina's MySeq machine has been like even kind of like the gold standard machine that people use as well, if you find from a lot of publications. So in general, amplicon DNA sequencing, basically, you just amplifying the conserved region from the environmental samples. And this is where people trying to study into the biodiversity and also the abundance of species. So in general, so of course, like what I mentioned, you might be getting samples from a lot of different area, different source. When eDNA first started, it always starts with the marine eDNA research. That's the most popular, and in fact, a lot of publications are focusing on the marine eDNA research. So on the marine side, on the water side, be coming from seawater, river water, stream water. So basically, people used to detect water quality or assess environmental issues. So which if you look into on the practical part or the applied side of the utilization of this eDNA work, it could actually be a very good guidance for the restoration effort from the government or from the particular department, and basically from the environmental impact of industry. So what example that actually I can share is actually in some of the prefecture in Japan nowadays. So they do in Japan, as you know, they have a lot of manufacturing plant around somewhere outskirts of in some prefectures, but there are already some regulations or laws that's in place in specific prefecture that all these companies or manufacturing plant are they are mandated to provide or to perform environmental assessment work around their factory in regards to the wastewater that they are discarding, and then how does that impact, or will that actually impact the environment around the plant, which might have some residential areas around them, or even some national parks, or even forest area or some park around the factory, for example. So this is where environmental DNA research has been a really popular area, specifically in Japan. And in fact, this company or manufacturing plant actually engage a lot of academic researchers to conduct eDNA research for the environment or the surrounding of their factory. So this is also one of the example applied, a real example that we see in some countries. And of the fourth point, same thing, some countries are actually using eDNA approach to detect and prevent the transfer of invasive species. So later on my slide, I will have a very nice example from Australia, that how have they utilizing eDNA approach to monitor invasive species at their ports. Okay, and then basically looking into to strengthen the marine biosecurity in Australia. And also on the endangered species part, so this is more on the conservation part. So I believe every country might have some very precious species of animals or even plants that they have in the country that's really, really important to the country. So this is where, again, eDNA approach is being utilized by not really harming the actual animals, but not really implementing the need of hunting down that particular animals to count the number of them. So this is where the beauty of the advantages of eDNA can come into place, and is one of the most, I would say, best approach to really help to manage endangered species in the country. So I mentioned a few examples, right? How eDNA has been used for biomonitoring. I mentioned about on the marine, seawater, or river. So as you can see here, so this is a good just overview of how eDNA is being utilized in different area. And as you can see here, the top 4 square here, there's marine, equestrian, freshwater, terrestrial, and then the different type of species or microbes that people are looking at from different source of samples. So this is where I would say a good capture of some sample source that people use eDNA metabarcoding for biomonitoring. All right. So next, I would like to, like what I mentioned, there's a lot of interesting work or research or initiative that's being done in our region, Asia Pacific, Japan specifically, in the eDNA space. So first, the one that I just mentioned, Japan is definitely one of the most advanced country in leading the eDNA research. So what I'm showing here, MyFish, actually, so this is one of the approach, I would say, universal fish primers protocol for metabarcoding that's established by our Japanese researchers, and to really identify the fish species from river, from the sea. So this is actually established by a group of scientists where they actually have a society. They call it eDNA Society Japan. So for this eDNA Society Japan, they are a very open society that actually encourage a lot of collaborations so basically the aim of the eDNA Society is really to establish maybe a SOP for eDNA research. But of course, it's not an easy task because, like what I mentioned, you have so many different type of sample source, different type of samples, the different type of organism that you want to look at. So it's actually a long, huge project for them to accomplish. But so this is the MyFish protocol is one of the very first that came out and being published, and basically is a openly shared protocol for other researchers to utilize as well. So you guys can look up a MyFish, and this is where all the different publications that you can see. So again, with the MyFish approach, they also have the pipeline as well. So the bioinformatics pipeline, or the database that is openly to be used. So same thing, you just Google it, MyFish pipeline, you will pop up, and basically you just can, it's as simple as basically you can plug in your NGS data, and then they will just help to screen through the database that they have. And then yeah, I mean that's one of the easily accessible pipeline that's available. And also, of course, these are all the different publications that I just captured and bring out for people who are actually doing on fish, for fish detection. So there are actually a lot of different references that's been established within the fish space using the metabarcoding approach. So moving from fish, of course, there are people working on mammal as well. So some of the one of the publications that's happened in Sabah, Malaysia, Borneo, but it's actually executed by Japanese researchers on the mammals, the tropical forest mammals, as well as some terrestrial mammals from the fresh pond water. So there are actually a lot of publications about eDNA and on different species and different approach that you guys can easily just Google Scholar and just search for it, and to start some reading and some understanding about the latest trend that people are doing. And also there are some studies on bird as well. I won't go into the details of this, just to bring up and highlight to all of you, and amphibian as well. And of course, I mentioned about earlier in my slide, COVID. So eDNA has been used on the wastewater surveillance as well. So this is one of basically taking the water and then trying to sequence and see what are the, if COVID actually exists in the wastewater or the drainage that could pass and spread it out to the public. So this is one of the study that Japan has done. I think a lot of different studies, I think in New Zealand as well. So working on disease surveillance using DNA approach as well. All right. And so this is something that I would like to spend a little bit more time to highlight. So last year, actually, 2023, there are two major eDNA conferences that happened in our region. So one of it is the the very first Australia-New Zealand eDNA conference that happened in February, Tasmania. So on this particular, my presentation, as I mentioned, is focused on, I want to share a little bit more on the monitoring side. So this is one of the examples that actually pluck up and share, want to share with you guys, is about how they actually use. So there's this particular presentation that's very interesting that use pollen collected from bees to detect and identify any invasive species. So this is done by a institute called CSIRO. So basically they are using the pollen that collected, and they basically collect the DNA and try to extract the DNA, subsequently NGS, metabarcoding approach, and then try to identify the invasive species that they don't want to, that they are preventing, basically. So as you all know, Australia is a very strict country. So basically you are not able to bring any meat, food, or even fruits or vegetables in. So one of the main reasons really to prevent any invasive species of microbes or any organism that could impact the be it livestock or vegetation or crops in Australia. So this is one of the very interesting way that they are using, actually basically based on the pollen collected from bees, and then subsequently to detect it if there's any invasive species. So these are some of the publications that they have shared as well. And the second one is actually on something that I've touched on as well, along the line of invasive species from the marine biosecurity. So before we go into the specific example, so why does marine biosecurity matter, right? So basically, there's some study, like in terms of what's the impact of invasive species that everybody's keep talking about. Oh, the invasive species might be coming from sometimes, like we fly from countries to countries. It could be from the food, it could be from the clothes that we bring in, and in these regards, actually is in terms of the marine biosecurity. So for example, when there's a ship or when there's a vessel, let's say going into Indonesia, park at the in one of the Indonesia port, and then subsequently a lot of time they'll have this reserve water that they use, they call it ballast water, that will release into the marine space of Indonesia, and this is where the invasive species highly likely will be introduced to the marine area, and then subsequently kind of affect the whole biodiversity in the country and in the marine space. So basically, invasive species, there's some study that is the second most important cause of biodiversity loss across the globe. And if we look into Australia specifically, so there's more than 250 billion per year benefits from Australia assets are vulnerable if invasive species manage to penetrate, and up to 13 billion per year potential damage cost if there isn't any biosecurity control in place. So eradication measures, so meaning like after you identify there's invasive species, you're trying to salvage or try to save anything that you have, or to prevent it at that time, it actually will cost even more, like up to 20 million, and success rate is only 2 to 20%. So this is why kind of like these few points kind of highlighted the importance of monitoring, biomonitoring or biosecurity monitoring before all the damage happen, right? I think this could be not just, I mean, Australia will be just is a very good example to bring up the a use case scenario, and I believe this will be applicable to all different countries as well. So of course, early detection and management is the key, and then a specific example similarly they presented at this eDNA conference as well from Queensland Seaports facility or department, if I get it correctly. So basically, they actually formed this eDNA surveillance program or team that kind of carry out the early detection for invasive marine species and trying to manage biosecurity threats if there's any in advance or before anything happens. So basically what they do is they have involved 5 Queensland port authorities, and then also together with 16 trading ports. So basically, as you know, there's a lot of ports around Australia and a lot of vessels coming in and out along the coastline. And then this is just for Queensland. So because probably some of you are aware, like Australia, they do have a central government, but a lot of all these state relevant program or regulation is still pretty much relying on the particular state's government or authority. So this is kickstarted by the Queensland state. And then this is one of the program that they have kickstarted. So basically they have this program or setup for eDNA surveillance. As you can see here, together with 16 trading ports, as well as 12 of which are located adjacent to the Great Barrier Reef World Heritage Site. So what they have is, so basically is trying this particular program, actually what they have in details, I did put it here, is basically at every port, they actually have they call it like a platform that actually enable the growth. It's actually a platform that actually enable, if there's any growth that happen within that particular platform that is placed underneath the seawater, then they will have a regular extraction or capturing of that, whatever growth that they capture on that particular platform, and then they'll harvest it, and then they'll extract the DNA, and then they will, same thing, do the meta-barcoding approach. So basically they're using our MySeq, and then to see if there's any presence of invasive species or microbes that they don't want to see at the marine space. So this is where they're doing, and I think they're doing it on the monthly basis. So at these all different ports. And I think there's already planned to kind of expand it out, and then in other states of Australia, they are planning to implement some similar thing as well. So this is one of a very good example of how eDNA surveillance has been implemented by the government, and in terms of on the marine biosecurity part. And the next thing is, after I've shown you guys some of the examples and some different applications that people use using B, and then using at the, what they do at the port. Something that I would like to share is actually the what I mentioned, what's happening in our region, Asia Pacific, Japan. What are some societies that are present, and also some conferences perhaps that some of you guys would be interested to join, to really to have probably some very good discussion with all the rest of the eDNA researchers. So hopefully with this particular segment, we'll give you guys more ideas, like actually who can you guys talk to, which particular society that you can reach out to. So something that I will share in the next few slides. So what I mentioned just now on the MyFish pipeline, if you still remember, eDNA Society Japan is one of the very first eDNA society that's being formed in our region. So basically, something very interesting about eDNA Society Japan is they are actually not just formed by eDNA researchers from academic, but they actually open up their membership to commercial companies. If you remember what I mentioned just now, a lot of plant or manufacturing plants, or even those commercial companies, are mandated to perform eDNA assessment, environmental assessment kind of work, or eDNA testing. So these people are actually part of are being invited as part of the eDNA Society Japan as well. So in short, it's actually a very good ecosystem, or it's a group of people that come together that would love to, that has the passion in implementing eDNA approach. And of course, they are also, like what I mentioned, trying to establish something that could be utilized by everyone, something easily to be implemented. And of course, now the key method that they're using are still meta-barcoding. So on the eDNA Society Japan, if you just Google it, you'll go to the website, and it's quite nicely done. Basically, you have a Japanese and English language as well. So this is where I kind of screenshot it out for you guys to see. One of the, on the top left-hand side is actually the eDNA sampling manual that they actually shared. So it comes with a really nice video as well, in terms of, I think this is mainly on the seawater, if I'm not wrong. And then they actually teach you what kind of filters that you need to use, what are how much of, how many liters or gallons of water you need to run through the filters in order to get a good adequate amount of DNA to perform your NGS. So all these has been documented nicely. So I will encourage all of you to go and take a look. There's an English version there. And also on the right-hand side, there's video as well that's being shared on the website as well. So I think on the website, so every year, eDNA Society Japan will have their annual eDNA meeting. So sometimes they will have their minutes translated to English, but it's not that updated sometimes. So, but I would say that's still very nice to visit the website to take a look at the manuals as well as the video that they have. And the bottom part is actually another key conference that Illumina has been involved with. So working with eDNA Society Japan, we actually have this joined the eDNA International eDNA Conference. So just to let you guys know, so for the past years, eDNA Society Japan have their annual meeting, but it's always close to, it's a Japan internal meeting, which they only invite Japanese eDNA researchers. So last year, this is the very first one they actually open it up and invite a lot of basically international eDNA researchers. And then, yeah, I think this will be they will organize this every 2 years. So it's a very good conference that a lot of people will share a lot of interesting stuff or new stuff that they're doing around the environmental research. And this is also one of the conference that I've heard a lot of interesting work done around eRNA. So how they actually handle the RNA. So how they use RNA instead of DNA. And then, yeah, of course, with some specific reason. So like there are some argument as well, like DNA, like what I mentioned, it could be detectable up to 48 hours. So there's some argument to talk about it might not be a good reflection or representation of the real situation of that particular environment, because it's been there for so long and then it doesn't reflect like the actual situation. So that's why people start to think of, can we use RNA instead? Because RNA will degrade. So whatever we try to get from there is something, the real situation that we want to see. So this is one of the, I think a key, one of the research that's being presented at this eDNA conference in Untuk, Japan. And in fact, it's done by a commercial company. So something I would like to share is, like what I mentioned, industrial companies are also part of the members of this eDNA Society Japan. So something that I would like to share here, the list, probably some of the names here that you might be familiar. I believe the last one, Toyota. So it's interesting that actually these companies, they are big manufacturing companies which has their own R&D arm. So this is where they actually, all these 4 sites are actually, they own their own, they have, they actually purchase their own NGS system from Illumina to perform the eDNA testing or work that they are doing. But of course, because it's kind of considered confidential of what are the things that they are doing, so we are not very clear about the actual study or R&D work that they are doing. But at least we know that it's all relevant to environmental study. And something about Toyota as well, something that they are doing is actually on their aircon system. So they are doing some interesting work around air DNA. So basically DNA that's captured from the air, and then they are trying to implement it, I think some R&D work around their aircon system in the car as well. So the third one, Cow, I'm not sure if you guys have seen this brand before. So they actually produce a lot of toothpaste, detergents, toothbrush, so a lot of the cleaning reagent. So same thing, this company actually is the one that I mentioned are performing eRNA work. So very interesting work as well. So I think they are doing some eRNA work from the wastewater that the manufacturing plant discarded. So yeah, so a lot of, and the top 2 are engineering, big engineering company. Some are like big shipping company as well. So I think some of them are doing some environmental research on the seawater as well, after I think they discard some wastewater to the sea as well. So very interesting things that's happening within the eDNA space in Japan. And from Japan, let's move on to Australia, New Zealand. So this is the second, I would say, after eDNA Society Japan. Southern eDNA Society Japan just formed last year. And actually they call it Southern because it's formed by a group of researchers from Australia and New Zealand. And if a little bit different is there isn't many, commercial companies that's part of this society, but there are a lot of eDNA service providers that's coming from Australia and New Zealand that are part of the societies. So what they are trying to do is, again, promoting science and industry collaboration across Australia and New Zealand to advance best practices, like what I mentioned, SOP for eDNA method, and subsequently adoption in government and private sectors as well. So Illumina, we have been engaging with them since day one, and we are actually the education partner with them. So sorry, I'm going back a little bit. So the port eDNA surveillance at the port, the eDNA surveillance monitoring at the port in Australia, that particular project is actually presented in the Southern eDNA Society the Tasmania conference that organized by Southern eDNA Society. And the next thing is something very interesting as well that I would like to highlight along the environmental research as well. So basically, Illumina has this collaboration with this particular foundation. So it's a non-profit foundation. It's called Minderu Foundation. It's a philanthropic organization in Australia. The boss is one of the richest guy in a billionaire in Australia that involved in mining business. So he has a lot of this. So he established this foundation, this called Minderu Foundation, and one of the arm of the foundation is actually to look into the biodiversities in the ocean. So as you can see, the second point there's this Flourishing Ocean Initiative that they aim to expand the marine protected areas, look into biodiversities, and then for now they're specifically focusing on the Western Australia. So there's this program called Oceanomics program that we work with them very closely, and in fact they are the very first eDNA researchers that bring our NEXIC 2000 onto a ship, onto a vessel, and they perform the NGS, the basically everything, the whole workflow on the ship. So they have done a trial. So this is not really super updated because they have done initially a trial voyage on our NEXIC, and subsequently they are very happy with the results, with the onboard, on-vessel sequencing work and everything, the results. And then subsequently, they actually purchased a new NEXIC 2000 to be placed on the ship. So the image that you see here is a upcoming vessel, a really luxurious vessel that the CEO of this Minderu Foundation planned to build from scratch, where you can see there's a helipad there. So there will be a mobile lab on this vessel for them to perform eDNA work and NGS on the ship. So one of the main reasons to have the machine on the ship is that, as you all know, that if you guys are doing some marine research, every time you need to get samples, you might need to take a boat or vessel out, and then it might be a few days time, you collect the samples, and then you come back, and then you perform extraction and so on and so forth, NGS work at the lab, right? But with the capability of this on-vessel facility, sequencing facility, basically they can perform the sequencing immediately. And if the results that they are getting is not something that they want to look at, perhaps, for example, the samples that they get is from this particular depth, let's say 50 meters. Maybe they are not seeing what they want to see. Maybe they want to get samples from even deeper seas, like maybe from 100 meters. So without having to go back and get the results and check, oh, we need to go back again, it actually saved them a lot of time. By maybe the next day or the next few days, they can straight away, since they are already out there on the sea, they can straight away get the samples immediately. So this is one of the consideration or the beauty of having a vessel with sequencing facility. But of course, not everyone will have that capability to do it, but this is just to highlight the flexibility of NGS, as well as even for Illumina machine. It might not be as small as a PCR machine, but potentially if you have a decent area, we have already customers that implemented that and actually brought the whole machine onto the ship and perform NGS successfully. Okay, this is just an example that we like to bring out and share with you guys, that something that's happening in Australia. So a quick one. I know I'm over time. I just want to show some of the video that we have done with Minderu. Maybe I'll start with this one.
My name's Angela Healy. I'm a Senior Field Application Scientist with Illumina, based in Perth, Western Australia.
My name is Priscilla Gonçalves. I'm a research scientist and the head of operations of the Genomics program.
My involvement in the Oceanomics program is that of Illumina application support. So I'm helping to install new instruments, train the Oceanomics team on the technology, a little bit of troubleshooting and consulting on new assays.
Our work is focused on marine genomics and environmental DNA. When considering the technology required for the Oceanomics program, we looked for partners that could not only provide access to world-leading technology, but also think through and embrace the challenges and the risks with us.
When Priscilla first engaged us to ask if it would be possible to use a sequencer like the NextSeq 2000 on board a boat, we really didn't know if it was going to be possible. We were actually all pleasantly surprised when we performed that first run, and the Oceanomics team are the first to have attempted it.
Illumina is a market lead in short-read sequencing technology. Not only that, but they are also working with us to tailor their products and services for eDNA applications.
Genomics is at the core of what the Oceanomics program is trying to achieve, and through providing the best sequencing technology for the job, we're helping to drive their important work. The NextSeq 2000 is perfect for the sequencing at sea, for the eDNA samples, helping to monitor the health of the ocean in real time. My favourite part of this program is working so closely with the Oceanomics team. They really are an incredible group of scientists that are passionate about the work they do. It's very clear. So it's really nice to be around that. It's inspiring for me as a scientist as well.
The challenge of returning the ocean to a flourishing state is too big and too urgent to be tackled by organizations in isolation. By bringing the lab, both wet and dry labs, to the field, we are bridging the steps and increasing efficiency between sample collection, processing, and analysis. With more than 3 billion people relying on the ocean for their livelihood, we need effective management, conservation, and restoration plans to mitigate the current and future impact on marine wildlife. Knowledge is power. The more we know, the better we can act.
All right, I know I'm running out of time, but I think this video is something that I really wanted to show you guys. So basically what happened, as you can see, everything has happened within that container with the sequencing machine on it. And then we have a FAS that went out to the sea with the Mindaru team and actually performed the sequencing work. All right.
The ocean is critical.
So, sorry. Yeah, I want to quickly, I know time is up. Let me quickly finish what I still have. And then another exciting example that we have just recently, late last year, is this initiative called Ocean Race. So there's this one of the customers that from New Zealand that worked with one of the race sailboat, one of the sailor, to really use the sailboat to collect sea samples at different part during the whole race. So the race is called Ocean Race. You guys can just Google it. And in fact, then they have a small device that they installed at the back of the racing boat, and then subsequently to extract the DNA and then, collect the DNA, and then subsequently to keep it in good condition, subsequently bring back to the lab to perform it. So it's just a good example how even sports or innovation that combined with scientific, basically a good collaboration that we can show that actually how eDNA can be implemented as well. So basically this is spearheaded by this Cultural Institute in New Zealand. Basically, they're using our MySeq. And then, of course, the ultimate goal is really to use the eDNA data to show how climate change is affecting the ocean biodiversity. So stay tuned for this one. We'll have a lot more collaborations that we work with Cultural Institute. And something that I want to share, last one, is actually from Illumina side. We actually, not really, I mean, a lot of time you guys might be saying, oh, you guys, Illumina is recommending this product, that particular product. But what I want to highlight is on the environmental DNA side, we actually built a landing page that is really meant for education and awareness purposes, where we put all the content that we have created, the interview that we have created with our customers, even as simple as introduction video, what is eDNA, animation video, as you can see here. So we put it all on the one landing page where you guys, in fact, everyone can access to it and take a look at it. And this is just a capture of what we have. And then also we have education video and infographic as well. And some icon letters are actually the interviews that we conducted. First one with Japanese customers, I think eDNA Frontiers. The second one is Australian eDNA service providers. And last but not least, I think this is the last one is actually the MyFish approach. The researchers that developed the MyFish approach. So all these are available for you guys to take a look and to understand like what other peoples are doing. And then last but not least, we also have put in some literatures as well as some webinars. So basically this is webinars organized by Illumina that we invite our customers to speak. And then basically once if you register, and then basically you can watch in demand as well. All right, that's all. And also sorry for running over time. There's so many things to share, but again, I hope you guys are happy with what I've shared today. So I'm not sure if you still have time for any questions.
Yes, thank you, Mr. Charilu. It is a very interesting talk. I think we learned a lot from you regarding the application of eDNA and also specifically for biodiversity monitoring. And I think we have plenty of time for discussion. And we have lots of questions in the chat box.
Okay.
But first, I would like to invite if there are any participants who would like to ask directly to Mr. Lu. Is there any question directly from the participants? Okay, I think we will start from the chat box. So we have the first question is from Mas Andika Prasetyo. I wonder what we need to consider on the library preparation stage in avoiding undercluster or overcluster when we're dealing with short fragment across different Illumina kit and Illumina machine. So yeah, what to do to avoid this undercluster or overcluster in Illumina? Do you have any comments on that?
Yeah, again, this is again like what I mentioned from the start is sometimes it's quite challenging sometimes to pinpoint how to like avoid the overcluster and things like that, because there are a lot of different steps that might impact. So again, I'm not sure what's the I'm not sure who's asking the question, but again, have they started? I mean, have they performed a run before yet, or this is the very first time, or this is the first issues that they are encountering, or because it's quite challenging. It's quite difficult for me to pinpoint, say, like how to do this because of this, because there's without understanding the whole picture, it's very difficult for me to give a very clear answer, and also understanding how's your experiment look like, what are you doing, what do you want to get, and things like that. So I'm sorry that I can't really give a really specific answer to that. But one thing is, I mean, I'm not sure for you guys who, let's say, if you're already very experienced in what you guys are doing, or you are already doing some eDNA work, feel free to reach out to us if you encounter any technical issues. Actually, we have a technical support people, my technical support colleague that actually can run through with you, pinpoint every step. So they can contact through email or phone call. You can just call them directly. But I think they are actually quite, as compared to me now, as in they will have more time to really pinpoint or run through with you guys what are the steps that you have done, and then what are the steps that you think that create the problem, and what's the problem, and things like that. And subsequently, even on the overcluster part, is it on the machine part, and how the preparation on the library prep, what has been done or what has not been done. So these are the things that I think for them will be better for them to kind of identify and address properly, like rather than me just giving a general comment. And so I would recommend same thing, reach out to us, reach out to Pandu as well. We are always here to help. And then just anything, just reach out to us. And I'm actually very happy to see what Rifki shared, the BRIN ELSA services that you guys have. I think that's actually a very good setup actually to perform eDNA services, from benchtop to even the NovaSeq. You want to do targeted, you want to do shotgun metagenomics. So it's basically you guys already have everything. Just need to maybe have a good proposal, and then subsequently to see what's the direction that you want to head towards.
Okay. Great, thank you. And then we have another interesting question from Ibu Febriana Sari. How far this meta-barcoding method can cover the unknown or new species and differentiate between them?
Yeah, so this is, I think, probably one of the disadvantage, I would say, for meta barcoding. So basically, in general, of course, the reason why we use meta barcoding, for example, is you need to, for example, let's say your project is looking to some fish species in that particular river, for example. So of course, the meta barcode you need to have is basically the fish species, the barcode that target the fish species. In general, maybe you can target like a particular fish species. So with that, actually, it will be very easy for you to look into it. But if you really don't have any idea, and in terms of particular new species, so a lot of this, it might go back to the foundation of your foundation or project in terms of how well studied is the particular species that you want to look at. And are there any references that's already available that you can utilize? And if the meta barcoding, so you need to have basically a good expectation of, if I'm using this meta barcoding, roughly what am I expecting? If something really new, novel, unreported by anyone, so I think meta barcoding might not be a really good approach for you. But of course, I wouldn't say it's a definite answer. Sometimes you might be able to caught by surprise. There's something really interesting that you want to, I mean, it's beyond your expectation that you can find out, that you can fish out from the barcode that you have. It could be something, but I would say meta barcoding approach generally is always good to have a prior understanding or information that you have, and as well as understanding of what are the references that you can utilize to really look into what you want to look at. So for really novel kind of species, it might not be a really good approach to utilize or to look at. Yeah.
And the next question is from Pak Ansari Maruzi. So he's interested in the statement that eDNA can help detect and prevent the transfer of invasive species and support biosecurity. So the question is, he's working on the development of methods for detecting adulteration species in medicinal plant raw materials from plant species, and how could application of eDNA could help with this topic?
Wow, the adulteration one is a very interesting area, and then I must say, until now, even so, in general, again, it really depends on how the foundation of that particular species, any particular references that already have, how strong the references and database that is already available that you can utilize. So one of the examples that I actually see from, if I'm not wrong, it's from Australia side, a few years back, is actually a company working on trying to use eDNA to test for looking into the different wood that people bring in. So basically, I believe, yeah, for Indonesia, probably you guys will have a lot of good quality of wood that fetch a very good price, and then subsequently, and then some of the furniture that's made by this particular wood, and then people will charge at a very expensive price, or even some really species of wood that is really rare and maybe controlled by country and not to and not supposed to do the deforestation and everything. So for now, I don't really see there's a lot of people are using on the plant side yet. How can it be utilized? I think this is something that I think is very interesting, that I've spoken to customers in Thailand as well. So same thing, they would like to utilize this as a eDNA approach to look into medicinal property from herbs as well, and things like that. So again, my answer is for now Illumina don't really have any specific solution for it. As you can see here now, what we have is basically, yeah, we provide the short read technology, and in fact we are not the one to actually really recommend a lot of different applications. This is where Illumina is very open to work with all the different researchers in establishing different applications or protocol. For example, the MyFish that the Japan customers has established, we work very closely with them. But on the the plant side is something that is still lacking from our side. But I mean, please feel free to reach out to us, and we can, in fact, maybe discuss with our field application specialists as well to see what do you intend to do and which particular, I don't know, species that you want to look at, and what's your ultimate goal as well. And then we can try to work together on this. But in short, I'm sorry, I don't have really a good answer to it, because I don't think I've encountered any specific implementation in that space yet for now. Maybe there are some R&D work or some research work that been done by academic side, but not that I know of yet. So yeah, potentially one of the good way that I would suggest is always, same for me, I always try to look for publications. That's the best way for me to try to look out for anything new, and then subsequently I'll try to work from there onwards. But again, feel free to reach out to us, and we can see how we can help you guys out. Yeah.
Okay, thank you. And the next question is from Ibu Ita. So the question is, by taking contaminated soil and using eDNA approach, could we know how many microbial species in the samples? I know we can, but I would like to add my question as well. So I'm working with the post-mining soil. So it is really difficult to get the DNA. I mean, I always have a low yield of the DNA extract from that type of land. And could you share some solution to increase the yield?
So that part is the upstream part, which is more on the extraction part. So I really don't have the answer for now. I might need to double check, because we do have a very interesting presentation in regards to post mining or the mining at the Australian eDNA conference as well. I need to dig that out, my notes, and potentially, of course, for me, I'm not the expert in this particular area, but what I can do is potentially to link up or to let you guys know that particular researchers who are working on that area. But in short, going back to the question, I think that will be on the upstream part, which is on the extraction part. So normally for Illumina site, we might not have a very good, so normally it's not that we are pushing this away, but again, we will refer back, we'll check with you guys what is the method that you are using, what's particular extraction kit that you are using, and then potentially basically some people are using some really traditional mechanical method to try to get the DNA out, and of course most of the time they might get a lot more, maybe slightly more DNA as compared to extraction kit sometimes, but it really depends on the method that you use. But in terms of when you get very low yield of DNA, besides that, are there a lot of other impurities still present, or is it because I'm not sure what kind of other optimization that you have done to kind of increase the yield? So yeah, but in general, I would suggest maybe this is something that you guys can check with whatever extraction kits company that you guys are working with, and then subsequently, then from our side, our technical support might have some information in terms of customers might have worked using with this particular kit that have maybe better results, and then yeah, so these are some of the recommendations that they can provide. But yeah, we are still not the best person to consult in terms on the extraction part. So maybe a little bit more information or details would be helpful in terms of what have been done, what have not done yet, or what are the things that you can try to, because I believe that there are quite a lot of soil kit out there, but I'm not sure, because a lot of people sometimes they'll even try to improvise and add some manual steps, extra cleaning or anything to try to increase the yield. But I'm not sure on that particular because I have to admit I'm not the expert in that particular area. But what I can do, like what I mentioned, in regards to the mining space, I can try to link you guys up with the researchers that actually presented on. I think he did something similar, mining and post-mining, and then trying to study the differences in terms of the microbial composition. And then if there's anything that bring in any invasive species as well and things like that. So this is something that they are doing as well. Yeah, but please, maybe later I can let Pandu share my contact, and then please drop me a mail so that I don't,
Yes.
it doesn't slip on my mind to dig out my notes. Yeah.
Okay, that would be great. Thank you. And then the next question is from Ibu Istiana. So, is eDNA from the air can be used for the analysis of the biodiversity of the plant species? And
Wow.
also in detail, what is the best method to collect the DNA from the air? And also, how about the biological replicates? Do you also need to consider the biological replicates? I'm not sure to do that.
Yeah, so basically for
Yes.
the air DNA, from what I've spoken to our customers, actually we have customers in Singapore, and then oh yeah, something that I didn't share on the air DNA part. Actually, there'll be one specific air DNA conference that will be happening in Australia sometime May in Canberra. So this is also organized by the Southern eDNA Society, but I'm not sure if they are opening that up as an international conference. Okay, this is just to share with you guys. So there's this upcoming air DNA conference. So going back to the question, so basically for air DNA, I think they are applying some specific filter. So for example, the one in Singapore. So in short, I've never heard any customers are using air DNA to detect plant species, but what I've heard so far is mainly using air DNA to check the air quality. For example, the one that they are doing in Singapore is on hospital environment. So basically, same thing because of the antimicrobial resistant issue and everything, One Health issue. So this particular PI is trying to work on air DNA, looking at the aircon filter system and that they have in hospitals. So basically, they are trying to study what kind of filter, let's say, is good to block specific harmful microbes or bacteria or viruses. And then basically what they get is from the filter, they're trying to extract the DNA from the filter. So main thing, I think, from what I've heard so far, from what I know so far, they are mainly using filter. But in terms of implementing it to detect plant species, I'm not very sure if the sensitivity-wise will be that robust until that, yeah, you can actually, I don't know, if a wind blow past and then you collect the air and then you know what kind of plant is there. I don't know, but I won't, I've never heard anything about that yet. Yeah. So air DNA so far I heard is all about filtered air and then trying to look into microbes, mainly microbes that are present in the air.
Okay, thank you. And then the next question is from Ibu Andina. So the question is eDNA could predict the population of fish in the ocean? I mean, you already mentioned that you have specific primers before in the presentation, but is it is it cover all of the fish species?
Not all. So basically, one of the things that I think probably eDNA, when meta-barcoding approach could do is, I would say another maybe disadvantage of meta-barcoding approach is you could know the abundance. So basically you roughly know there's a lot or there's a little, but you can't really pinpoint down to like the number of the particular quantity of that particular fish, for example. So that's one of the things about meta-barcoding. And in terms of the MyFish primer that is being established, targeting the fish, I don't think it's all the fish, but I think there's a particular taxa or particular group of fish that they can identify. So basically that is established based on from the marine samples or the marine space or the ocean within Japan. So of course, different marine space or different sea, different ocean might have a different type of species of fish. So you might need to double check on that. Like does the primer or the barcode kind of can kind of capture or amplify the species that you want to look at? Yeah.
Okay. And then the next question is from Pak Ahmad Sofyan. What is your suggestion to preserve the environmental sample, in this case, rumen, cattle, for a long time period? So, I mean, you have a good quality of samples for later DNA extraction, I guess. Do you have any comments to preserve the samples?
Sorry again. Sorry, I lost the first part. Okay. Sorry,
okay.
Zara, can you repeat again?
Yes. So Pak Ahmad Sofian is asking about the preservation of the environmental samples. So I guess when you need to store the sample first before you do the extraction, what is the best method that you could do to preserve your samples?
I think as long as, okay, so I think there's a lot of different, this is a good, I might not have the exact media, but I think there's a lot of different media that people use for. I think if it's for DNA wise, I think it's pretty standard. I have to admit that I kind of lost touch with the real experimental part of what are the reagents that I've used before to preserve the DNA. But even for RNA, like there's DNA later, RNA later. So there's a lot of different reagents that are available. But one thing is, just slip my mind. What's the key thing? And sorry, Zara, you mentioned what kind of sample is that? Room?
I think it's from rumen cattle. It's a rumen.
Cattle?
It's yes, like from the probably intestinal and also like from cattle. Rumen.
Okay.
Yeah. I'm not sure because for the soil, for example, when you go to the sampling site, you have this DNA or RNA preservation solution. Usually, I bring this, but is it also necessary for the rumen cattle samples? I'm not sure. Because microbial diversity is really flexible. Yes. Like when you have temporary aspect, it could change also, I guess.
I'm not sure on the rumen samples in this regards. Yeah, I probably need to get back to you on this one, just to make sure that I don't anyhow give a wrong answer on this one. But like what you mentioned, there's a lot of preservation solution that we have or we can use. Just that I need to double check on this one. Yeah.
Okay.
I'll just put it down.
Sure. And okay, I think we could move forward to the next question from Ibu Salma. This is so she's asking if there is any guideline provided by Illumina for ideal laboratory setup, specifically for NGS workflow, for example, using the MySeq or NextSeq platform.
Sorry, in terms of the guideline for?
The guideline for the setup in the laboratory for the platform of Illumina, for example, NextSeq or MySeq. Is it provided by Illumina, or you just, I mean, you could always consult to Illumina, right? If you would like to develop this type of lab, I guess.
Yes. So in terms of which, any guideline for the library prep in terms of the...
Yeah, for the setup in the lab, for the platform, I think for the workflow also from extraction, and then you do the library preparation, and then you do the sequencing. Is there any guideline from Illumina?
So like what I mentioned, specifically for eDNA workflow, we don't really have like a standard guideline. But a lot of times what we would do is we would refer customers to a specific publications that utilize Illumina's kit or Illumina solution or system, and then that will be the preferred way that we always will ask customers to look into, and then also to optimize accordingly based on the type of samples that they are working on, if you are talking about the whole workflow.
Okay. And then I think we have very general question at the beginning. So one of the participants, Ibu Rohani, would like to know the kits the extraction kits for bacterial and fungal species, specifically from the soil samples. If you have any recommendation on the best kit from soil samples, maybe you could share to us.
Okay. Yeah, for now, I don't have the information, the specific information of the recommended kit. But let me take this down, and potentially I'll double check with my tech support in order to provide a good recommendation. I think we do have some recommendation that customers can try, and then if that doesn't work, and then preferably can go back to the extraction kit provider to kind of discuss about the details in terms of how to optimize it. Yeah.
Okay. So I will move to the next question also from Ibu Penny Wahyu. Could you use eDNA to get some information regarding the how do you call it, like the cattle, if you would like to search for the cattle with the beneficial traits specifically for humans, could you also do some kind of eDNA analysis for that? How
Beneficial
so,
traits
like,
from cattle.
yeah, I think it is more related to the utilization of iScan, because with iScan you could like try to, I think eDNA is not really the best method to select for the beneficial traits for from animals, right? I think it's more like the application of iScan.
Yeah, those are like the markers that the beneficial markers that you want to genotype for. So potentially that's something you use for use the iScan for. Yeah.
Probably, yeah. And then I think it's I'm not really asking this question before, but this is also from Ibu Istiana. What is the universal primer available for a plant metabarcoding? Is it a lot of primers available for plants?
What particular plants that she's talking about? Similarly, for all these, I might need to defer to my technical support specifically in terms of particular specific like the universal primers that they are looking at. Because like what I mentioned, as compared to fishes or even other mammalians or other organism or species, plant is I would say a lot of studies, of course, more well established already for especially for fish or marine side of that kind of organism. But plant side, definitely there's a lot of publication as well, but we need to double check on that if that is something that's suitable or something that what she's looking at. Yeah. So we need some more specific information in terms of what she's looking at, and then subsequently we can potentially point her to the right direction. Yeah.
Okay then, and then I think we have one last question, this one is really specific. So this is from Pak Amri Prasetyo. So he's working on yeast diversity, and then he's looking into the diversity of the yeast using the primer of 18S ribosomal DNA. And then he found out that there are 51 species of yeast, and he would like to know if you have any experience working with yeast diversity, but he's also not really mentioning what type of samples that he's working. I think we need more information, right? 51 species.
Thanks for going. In general, and I have to explain. I mean, for me, I mean, I might not have some really specific answer to your question. I know you guys have some really burning questions about what can I do for my project for this particular thing or for that particular thing. Offhand, I have to admit that I don't really have all this information readily available that I can provide advice or suggestion. So that's why I would really encourage you guys to reach out to us, reach out to Pandu in terms of what you want to do. We are more than happy to assist you and happy to help you or to kickstart your eDNA work and really utilize what you guys have at BRIN. And then even for now, we might not have the best answer or the right answer, but same thing, we are happy to really work with you guys to to move together and to progress along to see how we can establish, like what I've shared in Australia, in Japan. These are some of the things that we work with customers closely, and we're happy to see some nice publication being published. And then similarly, we are hoping to see we have Indonesian researchers over here really kickstarting really nice work, because you guys have the, I think, very nice setup already. Subsequently, it's really based on the resources, the environmental samples that you guys are already working on, and how can we bring that together and really make full use of the data that you guys have. And then Illumina is more than happy to be part of that and assist you guys along the way. Yeah.
Okay, thank you. I think that's all the question that is from the chat box. With this, I would like to invite if there is any other question from the participant, or we could just, I think it's enough. I think we have plenty of question and answer. Thank you, Mr. Terilu, for answering and discussing with us. And yeah, I think we have arrived at the end of today's webinar. Pak Rifki, maybe have comments before we close the webinar for today?
No.
Okay.
Thanks,
Right.
everyone.
Yes, thank you. And I think I would like to remind you to fill the presency. So it is shared in the chat box. So for those who is not really, yang belum mengisi, mohon dapat mengisi ya Bapak Ibu. And also I think for the last session, we could take a picture together. And Mbak Rizka, apakah bisa minta tolong Mbak untuk foto?
Ya, bisa Bu.
Oh yeah, baik Bapak Ibu. So I would like to invite all of you to open your camera, because we will have a photo session for today's webinar. So we have three we have three slides, and then for the first slides, maybe Mbak Rizka, could you take a picture from the first slide?
Oke.
Okay.
Untuk slide pertama,
Mhm.
saya ambil 1, 2, 3. Yang kedua, 1, 2, 3. Layar ketiga. Ya, sudah Bu.
Ya, terima kasih Mbak Rizka. Menginfokan juga ya Bapak Ibu, nanti ada door prize dari Pandu Bioscience untuk 3 peserta paling aktif pada webinar hari ini. Jadi ditunggu nanti emailnya dari Pandu Bioscience untuk door prize-nya untuk 3 orang ya Bapak Ibu. Baik, demikianlah rangkaian sesi webinar Lab Sequencing yang kedua hari ini dengan topik Trends and Application of eDNA in Biodiversity Monitoring. Mudah-mudahan ilmu yang di-share hari ini bisa bermanfaat untuk kita semua ya Bapak Ibu, dan juga membuka gambaran terkait aplikasi eDNA untuk biodiversity monitoring dan juga untuk pemanfaatan layanan ELSA untuk menggunakan platform sequencing yang ada di Lab Sequencing BRIN, diantaranya ada Illumina ONT dan juga ada MGI. Jika Bapak Ibu ada pertanyaan, nanti bisa disampaikan langsung ke pusatsequencing@brin.go.id, ataupun tadi juga Pandu sudah share email ya tadi di chat box juga. Jadi ada https://bioscience.com, kontak us. Tadi saya sudah di-share di chat box Bapak Ibu, jika ada pertanyaan juga langsung ke Pandu. Kemudian lastly, I would like to present my gratitude for our speakers, Pak Rifki and Mr. Terilu. It is such a good talk, and we have a fruitful discussion. And I hope that we could also discuss later in the future, because we are currently developing eDNA lab in BRIN. So it would be great if Illumina could help us in developing this lab later. So thank you, Pak Rifki dan Mr. Terilu for today's talk. And then that's all the time we have for today. Thank you so much for all of the speakers and also the participants. If you'd like more information, you could just feel free to reach us. Bisa kontak kami di Lab Sequencing, Bapak Ibu. And we will see you. We hope to see you again on the next webinar, of course, with more interesting topic. With this, I close the webinar. Saya undur diri ya Bapak Ibu. Thank you all for the participation, and then see you later in the next webinar
Thanks,
session.
everyone.
Thank you.
Thanks, Zara.
Bye.
Bye.
Yes, thank you.
Makasih Mbak Jara ya. Thank you.
Makasih Pak Rifki. Ya, makasih Bapak Ibu semua.
Partisipan, materi.
Thank you.
Bye.